Feel welcome.
Okay, thank you for the introduction, Bern. So this session on SNP panels is where we're going to go from our large genome-wide data sets that contain tens of thousands, if not hundreds of thousands of SNPs, and reduce it down to a panel that will have only tens or hundreds of SNPs. And some of you may be wondering why in an age where sequencing is getting cheaper, large data sets are getting easier to handle, and there's wonderful programs like dartR that we've been going through to help us handle those large data sets. Why would we want to be reducing the amount of data that we're using? So this session is going to go through where SNP panels can be incredibly useful and make sure that you're going to be able to select your SNP panels appropriately to address various genetic conservation research questions. So before I get into it, I'm just interested to put a little poll out there just to see who's currently using SNP panels or who intends to be using SNP panels in the near future with their upcoming work, just so I can get a feel for where the audience is at with that. Let me scroll down. Okay, so we do actually have quite a few people using SNP panels. Probably a biased data set, given the people tuning in are probably the ones more likely to be using SNP panels. But anyway, good to see a few are using it. So we've got people using it for individual ID, population structure seems to be the most popular, but also provenance and relatedness. A few other uses there as well, which I'd be interested if you want to pop that in the chat, what some other people are using the SNP panels for as well. All right.
I will monitor it for you.
Okay, thank you. Jump back into the slideshow. Okay. All right, so the aim of this session, I guess, is for me to provide you with the knowledge and the practical tools to be able to go from those large data sets to those more targeted panels. So, as I said, we're looking at generating SNP panels, marker sets that contain around tens or hundreds of markers. Some of the advantages of these SNP panels are that they can be reproducible. So it means you can have standardized results when you're working across various labs, and that may be the case that you've got labs in completely different regions or completely different countries even, but they can be generating comparable data sets. It can also be really good when you've got longitudinal studies. So again, you've got those comparable data sets over really large timescales. SNP panels can be incredibly cost effective if you've got high sample volume. So there is an initial startup cost that you need to put into developing the panel in the first place. But from then on, once you've got that SNP panel optimized and ready to go, it's a much lower cost per sample compared to genome-wide kind of SNP analysis. The main reason I'm using SNP panels is because I work with trace DNA samples quite frequently. So this is low quality or low quantity DNA samples, and that's obviously particularly appealing for a lot of conservation genetic research because it's often a non-invasive approach. So there's been examples of SNP panels used from scat samples where we're looking at host ID or host population genetics. Hair samples, feather samples are other examples of trace materials, and environmental DNA is the field that I'm trying to look at the moment. So can we get population genetic information out of water samples? So when we have these really trace amounts of DNA, we just don't have the quantity of genetic material that's required to be able to generate genome-wide data sets. So those SNP panels can hone in and target some particular loci of interest. SNP panels can also increase our resolution for particular genetic metrics, and I'll go into some examples of that later on in this talk. And another advantage is that it can provide a quick turnaround for some rapid management decisions. So the data that you get can quickly be put into pipelines and things to be able to make some really key decisions that might be required in a rapid turnaround. So some of the questions that SNP panels can be used to address in conservation genetics include things like assigning individuals back to various populations or management units, and this can tie in with detecting illegal trade in various animals. So I've got an example there of cheetahs. This work was done in some of the southern countries in Africa, where the trade of captive bred cheetahs is permitted so that they can move captive bred individuals into different locations. However, it's forbidden for caught or wild bred individuals to be moved. And so they have a SNP panel that they've developed that enables them to identify provenance of these cheetahs, so they can identify whether individuals are being captive bred or wild bred, and can then generate that information quite rapidly to be able to make management decisions if they happen to happen to capture a wild caught individual. It can be also used to track pedigrees, so similar, I guess, to that cheetah example, but this can also be great for looking at relatedness in captive bred populations. So where we may not know which individual the father is, we can use SNP panels to help identify fathers or mothers in some circumstances. It can also be great for translocated populations where you have individuals coming from two different source populations, and we can look at how much they're contributing to F1 or F2 generations down the track. IDing individuals is a common use of SNP panels, so that's something that's obviously required for close kin mark recapture type of analysis. And it can also be used to detect hybrids. So most of you will be aware that in conservation terms, hybridization can actually erode the genetic integrity of naturally occurring populations. So it is something that we want to be keeping an eye on in some circumstances. And then long-term genetic monitoring, so various population genetic type metrics like heterozygosity, inbreeding, or population structure. Along with these kind of population genetic markers that we use in our SNP panels, you can have some additional metrics that you want, where you have just one or two SNPs that you'll use to identify sex. So you might have your sex-specific loci in there to identify males from females. You might have candidate adaptive markers that can help you look at adaptation if you've got particularly key traits that you're wanting to monitor. You might have diagnostic SNPs that can help you assign individuals back to particular populations. So you might want to add those diagnostic loci in there. And then there might be certain phenotypes that you're interested in identifying as well. So those phenotypic markers can help enhance individual identification for forensics, for instance, or for other population monitoring. So you might want to be looking at coat colour or eye colour, or in agronomy, or when you've got selective breeding, some of those phenotypic traits are obviously going to be of key importance as well. So there's a few various markers that you can throw into your SNP panel. I'll just take you through a couple of examples where SNP panels have been used in the literature. So this example here is looking at hybridization, and they had 5 canid species that they knew hybridized together. So they generated a SNP panel that had 192 loci in it. And you can see from the bottom of this slide here, we've got a structure plot that clearly differentiates our 5 species of canids. We've got jackals, dogs, and 3 species of wolves there. And then we can see in our F1 generations, we can clearly identify the parental genotypes in each of those individuals with a 50-50 split. And then we've got our back cross individuals where, again, you can clearly identify the parental genotypes in there as well. And within this SNP panel, they did include a few of those additional metrics. So they had 3 phenotypic markers in there, which related to the coat colour, the nail colour, and the presence of a dew claw, because that can help identify wild canids from other individuals. The SNP panels, as I said, can be really useful for some trace DNA work. So this example comes from brown bears, where they're looking at fecal scats in brown bears. So they went out to a site and collected fecal samples from a whole lot of individuals and ran them through a 96 SNP panel. From that, they were able to estimate population size, and they found that their population size estimates from their SNP panel fell within the 95% confidence intervals of their capture mark recapture estimates. And they also estimated relatedness. So you can see in our box plot figure there that when we have a known parent-offspring pair, the SNP panel identified the relatedness coefficient of those individuals with a mean of 0.5. And similarly for the full SIB individuals, we've got a mean just over 0.5 as well there. So it did a really good job of estimating population size and relatedness. There were also some sex-linked markers thrown in there as well, so they could identify males and females. So there's obviously a whole lot of useful population and conservation genetic information that could be generated by just these scat sample analysis here. So when we're selecting our markers, there are a few important considerations that we need to take into account. So we're obviously dealing with really low numbers of markers, so there's a lot less room for redundancy. So we want to make sure that every SNP we're selecting is contributing information to address our particular research questions. To generate our SNP panel, it does require genome-wide SNP data, and it requires that data across multiple individuals that span the geographic locations, or at least the populations that you're going to be using it back on. So that's to avoid things like ascertainment bias that can really creep in and kind of interfere with the data that you're getting back. And also just to make sure that our loci are amplifying and giving us useful data across those populations too. So there's just an example here to show you the kinds of things that can happen with ascertainment bias, or when your SNP panels are not appropriately developed to test on the species that you're going to be looking at. So this example is actually again from that same cheetah SNP panel that was used to look at the captive bred individuals in southern Africa. So it was designed on this light blue population that you can see on the map. And when that SNP panel was applied across a subset of those species, we can see 91% of the markers come up polymorphic, and we've got 9% of them monomorphic in that particular group. When we then apply that same SNP panel to this population up here in the north, we can see that it wasn't actually designed to be used on that particular subspecies, and so we've got 9% of those markers don't actually amplify at all in that population. And so we've reduced our useful data set down to 82%. If we then wanted to go and apply it to a completely different species, so we can see it's been applied to lions and wild cats and other cat species there, we can see we get a bunch of markers that give us no amplification, but also these monomorphic loci just increase dramatically. And so we've got a much reduced data set that we'd be working with there. So it's really important when you're generating your marker set to have a good think about where am I going to be applying this, which populations or which subspecies or which species even do I want to be using it on, and make sure the information you're putting into it encompasses all those groups. And just a note on that though, if you do happen to develop a SNP panel for, say, this southern subspecies here, and then later on you decide, actually, I want to also use this panel across a wider area, you can expand your SNP panel later on to include additional SNPs at a later stage as well. So targeted SNP panels are going to address specific applications, and it's worth, again, it's having a good think about when you're designing your SNP panel, what are you wanting to use it for? Because if it's designed to address one specific question or specific application, it may not be that useful to address more broader analyses. It's got reduced representation, so some genetic signal may be lost. So obviously a small SNP data set is not going to help you look at things like adaptation or selection within your data set. Some subtle structure, population structure, may also be lost within it as well. The other important thing to consider is that it gives you a new measure of genetic diversity, or all your genetic metrics really. So comparing whole number outputs that you're going to get from your SNP panels back to your previous genome-wide data set's just not really something you can do. So you're working with a new ruler, and you kind of need to keep comparing like with like. As I said, your requirements include that existing genome-wide SNP data and with good geographic coverage. So just some notes about when to use SNP panels and when to avoid them. So it's best to be using SNP panels if you've got a large number of individuals to monitor and you want to be saving on cost. If you've got long-term surveillance so that you've got that direct comparison over longitudinal studies, and when you've got DNA samples that are low quality or degraded samples, SNP panels can be incredibly useful. But it's best to avoid SNP panels when you've got very small sample sizes, because it just as I say, that startup cost can be a bit prohibitive. If you need something like genome resolution, obviously a SNP panel is not going to give that to you. So adaptation studies is a key example of that. Or when you're wanting to monitor genetically distant populations, but the genetic data that you're using to generate your SNP panel doesn't incorporate that full genetic extent as well. So what we're going to be going through in the tutorial is a new function that's been developed in dartR, and credit to Bernd for developing that. So the versatility of this allows you to select a SNP panel from your data set, allows you to modify the number of SNPs that you're selecting. So that might be a cost-related issue, or it might be a service-related issue in terms of how many SNPs you're able to work with. And it also allows you to find the best panel to address either one specific conservation question, or there might be multiple conservation questions that you're wanting to address with your panel. So when we go through our panel selection, there's kind of a 3-stage process to it. So the first one is obviously to filter your data, just as you would with any kind of genetic analysis. So you're wanting to filter for the quality of the SNPs. So removing monomorphic SNPs, filtering for call rate, and filtering based on minor allele frequency. But then there's another filtering level that we're going to do specific to our SNP selection. So we're going to need to high sequence quality, and we're going to need SNPs that have surrounding areas that are going to be suitable for our primer design. So in some cases, the sequence length that we get back from services like Dart might be quite short. So although they'll normally give you a 69 base pair fragment, sometimes they are much shorter. So we want to filter out those really short ones that we may not be able to design primers on. The location of the SNP can also be quite important. So you might find that within your 69 base pair fragment, your SNP might be located at one extreme end or the other. And so you don't have enough room to be able to develop both a forward and a reverse primer within that fragment. Or alternatively, best option if you can have it, is to have a reference genome so that you can then assign those fragments back to a reference genome, and then the location of the SNP doesn't become so important because you've got a whole genome to work with to be able to identify what your sequences are either side of that SNP. So there is an alignment function in there, a blast function to link it back to the reference genome. All right, then we want to decide on our panel goals, and the function here is GL select panel, and you can choose a various method that you're going to use to help achieve your panel goals. So I guess broadly we've looked at 4 categories that we can address with our SNP panel. So if you want a panel to help you look at population structure, that's often going to benefit from selecting loci that help differentiate a population, so with high between population variance. So the loci that are selected from a DAPC are obviously ones that help pull apart those differences in population structure. So we've got one method that helps you select your panels based on the loci that it contribute most to a DAPC. There's also a method called PA high, and that helps you select loci with private alleles that have a high frequency. So in some cases they can be diagnostic of a particular population, or in other cases it's just that the frequencies are much higher in some than others, and so obviously that's going to help you discriminate populations. And monopop is the other method we could use there, where we're selecting monomorphic loci within populations. So those are the fixed loci within populations. If we're wanting to look at individual ID, as I said, that's something useful for close kin mark recapture. It can also be useful for parentage or relatedness. That's where we're going to benefit from loci that have high levels of heterozygosity, because they're going to be highly informative at each locus. So PIC, or the high polymorphic information content with high minor allele frequencies, is something that's often used for that. So we have a method to help you select data based on that. There's also a method known as PIC DART, which is a value you can get from DART, which is similar to PIC but based on presence-absence of alleles rather than the frequencies. If you're looking at genetic diversity or inbreeding, we've got 2 methods there. So there's HAF all and HAF pop, which helps you select the loci with the highest minor allele frequencies either across all populations or within each population. And if you're looking at genome-wide diversity, you can just randomly select your loci, just to give you an unbiased snapshot of diversity across the genome. And I guess this one here is going to have the metrics that come out are going to be most similar to your full data set, although there can be a lot of noise around that, and we'll kind of have a look at that as we work our way through the tutorial. The other way you can select genome-wide diversity is to use a stratified model. So similar to random, but we might say we want to make sure we're capturing loci that have a frequency between 0 and 0.1, and then we want an equal representation of loci that have a frequency between 0.1 and 0.2, and between 0.2 and 0.3, etc. So it just kind of helps you stratify your random selection in that way. And of course, you can also add in those sex-linked SNPs or adaptive markers or various other markers here as well. So that's the point at which you can kind of decide to add those ones in. Once you've selected your panel, you can then check how well or evaluate how well it performs against your full data set. So the function there is GL check panel. And so we can check that across 6 measures. So we've got our FST, so genetic differentiation, we've got inbreeding, we've got number of alleles, expected and observed heterozygosities, and effective population sizes. So this example here shows you we had a panel that we selected based on a particular method, and then we want to look at how good is this panel for identifying FST within our population. So along the x-axis here, we've got what our original FST was based on the full data set, and along the y-axis there, we've got our new estimates of FST based on our reduced panel. And you can see we get an R-squared value of 0.97. So we've got really good concordance between those 2 methods. But if you take a closer look, you'll notice that the absolute values do differ. So previously our FST was only reaching up just over 0.3. With this new measure now, we've got FSTs getting up as high as 0.4 and slightly higher. So the relationship stays the same, but the absolute values are going to differ, and we can see that this new panel can actually give us some increased resolution in differentiating between some of those FST values between populations. Once you've selected your panel and you've checked it and it all looks good and you're kind of able to address all of those genetic metrics that you're hoping to address your conservation questions, there's then a few additional steps to make sure that it's lab ready. So you've got to select your sequencing provider. They're going to have particular requirements with how you prepare your data. You've got to go through the primer design steps. So there's some important bioinformatics to be done to make sure you're avoiding primer interactions, because you're going to have a whole lot of primers that you're throwing in there together into the mix. So you've got to be avoiding all your primer dimers and other interactions. And then even despite the most perfect looking bioinformatics, you can still have things going on in the lab. So it's always going to need that lab testing, because you're likely to have some loci that over-amplify or under-amplify or just don't go well together with other loci for whatever reason. So there's a few optimization steps required in the lab for you to filter out loci and do some rearrangements as necessary and retest it until you've got your final SNP panel that you're happy with and that is all working well, that you can then use into the future on your samples. So currently, what are these SNP panels mainly being used for? There's a lot of genomic selection for crops and animals. So obviously with crops, we often have high throughput, so that those really high numbers make it cost effective to be using the SNP panels. In ecological applications, it's mostly used for large volume monitoring of close kin marker recapture analysis. It's also used when reference genome marker data quality, reference genome marker data quality are going to be super important to make sure your design is successful. And the cost of it is dependent on the number of markers that you're going to use and the required sequencing depth. And again, that's all going to relate back to your application. So the example that we're going to go through in the tutorial is something that I'm working on in my DECRA. So as I mentioned, I work with environmental DNA samples or water samples. And this is my study species here, this little guy, the red fin blue eye. These are one of Australia's smallest and most endangered fish species, and they exist only in a single location in Edgbaston out in central Queensland. So they exist in a series of springs. You can see a photo of it there. They're just like large puddles, basically. They're spring-fed puddles, so they do exist year-long. What's happened with these guys is when Gambusia have entered into some of these springs, or as soon as Gambusia into the springs, they seem to outcompete the redfin blue eye, and we find the entire population within that spring goes extinct within a matter of months. So what's been happening is as Gambusia have encroached on a series of springs, the entire population of redfin blue eye was restricted to just a single spring. So a series of exclusion zones were set up around some of those springs, and Gambusia were eradicated from the springs. Those exclusion zones to make sure that Gambusia weren't reintroduced into the area. And then a series of translocations have happened of redfin blue eye, so all originating from that one source spring, that last remaining population, and they've been translocated into these springs. And in many of those springs, they're actually doing quite well, but we're wanting to monitor the genetics of them to look at, are they maintaining genetic diversity? Are they differentiated from each other? If we're wanting to start a new population, where should we be sourcing individuals from, and all of that kind of thing. So this is as a long-term monitoring kind of method we're wanting to use it as well. So the current method for monitoring this species would normally involve a fin clip, but because these individuals are so tiny, you just can't get fin clips from them and have them survive in the wild. So the entire fish needs to be killed to be able to do genetic analysis. Obviously, as an endangered species, that's less than ideal. So I'm looking at whether we can use environmental DNA as an alternative to the destructive sampling that happens with these guys. So in order to get genetic data out of environmental DNA samples, I'm going to need to design a SNP panel. So the first question is to decide on the aim and the number of SNPs that we're wanting in our panel, and then we'll work our way through the tutorial to do those 3 steps about filtering, and selecting our panel, and then checking our panel. So, Bert, I don't know if you want to...
Okay, you can hear me, can you? Okay. Yeah. Okay. Cool. Now there are ample of questions. Contrary to Elise's kind of prediction, I didn't have any questions, and not to one, and she has 10. Isn't that unfair? But you go.
Okay. From your experience, is there a recommended number of individuals we should have whole genome sequencing for when designing SNP panels? Right. I guess it again comes down to your research question, and you want to have good representation of your population. Ideally you'll have 20 individuals from each population, and that's going to span the geographic range that you're wanting to be looking at. If you're working with endangered species that might have lower levels of genetic diversity, you might be able to get away with fewer individuals. But yeah, it's hard to say a hard and fast number on that. Yes.
Shall I read it? No, you
That's all right, I can read it. Thank you for the comments. How much does sampling design at the sample collection for whole genome sequence stage versus SNP calling stage impact the results? I understand both are important, however, sampling design and collection is difficult. Yes. Yeah, and always a challenge when we're working in the conservation space as well. I don't know if I have a great answer for that. Did you want to add anything to that one, Bernd? I mean, obviously, if you've got poor sampling design to begin with, you're limiting yourself right from the outset. The data you can generate is only going to be as good as the data that you've collected. Again, it's really going to involve playing around with your data to understand whether if you've done limited sampling, whether it's captured most of the genetic variation that it is you're wanting to look at.
Yeah, I guess the first question, what is the panel designed for? And depending a little bit on that, if it's ID of individuals, then probably the sampling at the WGS stage is not that important because, you will ID the individuals quite easily with a certain numbers of SNPs. Yeah, well, Elise disagrees on that.
No, I was nodding.
Okay,
yeah, absolutely.
And if you're looking for diversity measures, then you want to have a good capture of diversity that's out there. I just pretend this is answered.
You mentioned that diversity calculated from genome-wide markers versus panel markers are not comparable because we've got a different ruler, but are they still consistent? Yeah. So that R² value is going to tell you how that level of consistency, I guess. So yeah, you can still be able to differentiate most diverse from least diverse. It's just when you're looking at the actual number. So you've got an FST of 0.2 that doesn't equate to an FST of 0.2 that you had using your full data set. So just be careful when you're looking at the actual numbers, but the difference between them should be consistent. And that's what that check panel function allows you to do. It helps you look how consistent is this panel with my full data.
You will actually treat that quite well, hopefully, in the tutorial. So that should be clear. If not, please ask again. Yeah. Give me a sec. This is not answered. Oh, no, it did that. Sorry. Yeah.
When dealing with organisms that mutate or evolve relatively easily, is SNP panel still relevant? Oh, right, that's a great question. And Bernd's going to do some simulations on that. Hopefully we'll get time for some of the simulated stuff to look at if we generate a panel today based on the genetic metrics, genotypic information that we have in front of us on today's population. How useful is it going to be in 20 generations time if we're wanting to use it in longitudinal studies? So we can do some simulations to look at how's the population likely to evolve over time, and then how much is our SNP panel still going to be relevant in however many generations time? Yes.
It's really a great question because it tackles a lot of actually areas which haven't been super highly explored. There's a paper for coming out, and you select certain SNPs for FST and look for structure. It might be a selection of panels which are very inflexible if you want to monitor the change of FSTs. So even if it's great at the moment, it could not be a good selection. So therefore, simulations might help. You put a certain mutation rate on your panels, on the SNPs of your panel, and see how much it deteriorates, but you need to have a good understanding of the system to be able to do so.
And can we do SNP panels for eDNA, but how to handle markers that might or might not be appearing in more than one organism? Yes, so that's obviously a concern when you've got environmental DNA samples. You're going to have DNA not just from your target species, but from bacteria and any other co-occurring or related species that might be present. So we are working with developing primers here. So we've got a 20-odd base pair stretch either side of our SNP that we're trying to target. Hopefully that's going to stop it from amplifying a lot of other co-occurring DNA that you've got in there. Your biggest worry is going to be closely related organisms. So if you've got conspecifics, that's obviously something you've got to have to look at. If you've got genomes from those co-occurring species that you're worried about, that's something you can do a blast for your primers and see if it's likely to amplify in that. You could do a blast for those primers regardless across everything on NCBI just to see if it's going to be picking up anything else. So similar to what you'd be doing if you're looking at species-specific stuff. It's just we've got these species-specific markers that we're trying to develop across multiple SNPs in this circumstance. One of the reasons I've selected redfin blue eye as my study organism is because a lot of the springs, they occur in there, actually the only fish species there. In some of the springs, there are some co-occurring other fish species, but I know what those species are, and so I can have a look at the genomes of those guys as well to see if it's going to be amplifying it. So yeah, that is something you need to take good care of when you've got eDNA samples, and definitely something you need to think about.
Oh yeah, they never end. I guess we go through them, isn't it? Good.
When dealing with organisms that mute, we did that one.
Oops. Oh yeah, hang on. Let's check. Oh, my mistake. There's another one.
Is that the one we just did?
Did we?
Yes.
I'm confused now. Okay. Baseline change didn't do that one, is it?
No, I don't think so. If baseline genetic data for a species consists only of mitochondrial DNA or limited nuclear DNA sequences, can SNP data generated from dartSeq and analysed in dartR be compared with those data sets, or is a SNP-based reference data set required? Hang on, let me read that again. Baseline genetic data for species consists of only mitochondrial DNA. I'm not sure, is this question saying that dartSeq data contains only limited nuclear DNA? I'm not sure I understand the question.
Oh. I think, okay, yeah. Anyone from Dart who can answer that? We mark it as not answered and not answerable. I have to, because I would have thought you get mainly nuclear data when you do dartSeq. Anyone? No?
Unless the...
Seems like it would be very different.
Fun?
Seems like it would be very different.
Yeah, I would think so if that's the case. But okay, assuming you get that you have only mitochondrial data, then yeah,
okay. So we'll assume. All right. So we'll assume you've only got mitochondrial data or limited nuclear data, and you're trying to develop a SNP panel from that. Yeah. Again, the panel that you generate is only going to be as good as the data that you're using to put into it. So if you're just interested in identifying mitochondrial haplotypes and you've got that mitochondrial data there, then yeah, you could certainly generate a SNP panel to look at mitochondrial haplotypes. Your limited nuclear DNA sequences, it's going to depend what you mean by limited. So if you've got a lot of missing data across individuals and that data is missing at different loci, that can be very difficult. But if you've actually got good quality data but at only a small number of loci, then you can of course use that to help generate your SNP panel. Yeah, you've already got a limited number of SNPs, I guess, and you're probably just going to restrict it down even further when you go into your SNP panel. So it really depends on the data that you're starting with.
Okay, Florian is torturing us. What do you think? Use GL select to get your subset, yes, and then check that subset against the full data set. So yes.
A subset of the individuals.
Ah.
Yes. So no, you'd use GL select on the full suite of individuals. So the more individuals you can put in there.
Yeah, I would think so.
Better.
You would try to capture the whole diversity in the select one. Well, I'm not sure if I understand. Yeah, no, I get it. Yeah, okay. Yeah, no, we did that when we try to find out. Yeah, we used subsets and resampled or the subset of the data to make sure how much is basically the same, how much variation is there in the sampling. Yeah, you can do that. Yeah, makes sense. Yeah. Hang on, I just have to do it like this. Otherwise I get confused. Can we have one panel for several functions?
And can we have one panel for several functions or just one panel for one objective? You can absolutely have one panel for several functions. I'm not sure if we've included that in the tutorial, but you can, what we've been doing is with that red fin blue eye data, I'm wanting to address several different conservation genetic questions. And so you'll see when we go through and play with the data that some of the questions, like getting population differentiation, so FST is actually really easy to generate. So you only need about 10 or 20 loci to get really good estimates of FST. So you can select a panel that just gives you a few of those to help you get your FST, and then you can select another panel that will help you, add to that panel to help you look at some other questions like levels of heterozygosity or levels of inbreeding, for example. Yeah, I don't know what else you want to add to that.
No, that's exactly what we do. It becomes difficult and we don't have the answer. We're writing the paper at some stage and we haven't decided on the method because it's basically a set collection game you play there. You know, how many of the 10,000 SNPs, some of them might be good for answering the same question. So you use that. So our current approach is you select your 3 aims that you have and see what is the best panel and then find the panels which overlap the most in terms of loci. And that's a good panel, but we haven't validated that. So that this is a good question, but it's not easy to answer. So you have to basically combination of the check panel and the select panel functions. You can try to optimize your panel.
Yeah, I think that's where the check panel function comes in real handy because you can play around with the loci that you're putting in your panel and then check how well it works against the different metrics.
Jesus, there's hundreds of them.
I was wondering if there is any information on the number of loci that would be ideally informative versus too many or too little. Would we have to check that? Yes, again, so that's the function. When we select our panel, you can select how many loci. So you might start with just 50 loci. When you then go and check your panel, you might see, oh, the R-squareds aren't great. We've got a lot of noise around this one. What happens if I increase that to 75 loci or 100 loci? Does that help tighten our estimates? So yeah, that's definitely something that you can have a play around with, and hopefully these functions will kind of help you pinpoint what a good number of loci is that you can be working with.
Yeah, again, it's almost like here you can use R, basically what you would do, okay, I take 5, I take 10, 15, 20, 27, and you know, if you go downwards in numbers, how many you select, you will see that your estimate of FST and the ranking will deteriorate, and then it's your call, you know, how much information I want to retain. Obviously, if you use all the SNPs which you have, then you have 100%, but if you only use 10%, it's amazing. For example, some of the measurements like FST we found out, it's quite easy to get. Some other like inbreeding coefficient, if you want to monitor that, good luck. If you want to monitor effective population size, with the SNP panel, it seems to be really hard. Yeah, so there's some measurements which are easy and some ones are harder. And therefore, the number depends also on what you want to measure.
Maybe should we do one more question and then go into the tutorial?
Just 2 more.
Okay.
And we pretend no one else comes in, or we answer them at the end.
And can SNP panels be used as a fingerprint for variety identification? So I assume by variety, I guess you're kind of talking about it, populations or subspecies kind of thing, or variety of plant. So yeah, absolutely. In a similar way to where we can use it to identify populations. So long as when you've selected your SNP panel, you've selected it with that purpose in mind, and you've got the data that you're generating it from. So if you've got SNP data across the different varieties that you're wanting to compare, then yes, it should be able to address it.
And the final one.
SNP panels for highly threatened species can impact management decisions. Do you recommend cross-checking draft panels with other experts in the field before finalising your panels? Cross-checking draft panels.
cross-checking is always good, I guess. But,
Yeah.
but depends what you
Wondering
mean
how
with
you
cross-checking.
cross-check.
Yeah, exactly.
Yeah.
Because the experts in the field, what does it mean? And, you know, they don't know SNPs, I guess. Yeah, but they obviously have other recommendations. I'm 100% sure they know, you know, how easy it is to sample, how many you get, and how costly it is. So definitely you need to have this information, you know, because you want to determine how many SNPs do I get, how expensive it is. So if it's really easy to sample these individuals, then probably your decision how many SNPs you need actually it doesn't make sense. I'm not sure. Bill is interrupting me.
Quite difficult to coordinate different labs who are funded by different organisations without you even knowing. So it's very important to investigate that.
Yeah. Does it come through? No. Okay. So Bill was saying basically it can be really difficult to coordinate different labs, but you know, as far as I understand the question, obviously it's good to cross-check, but I'm not sure how much they can know. You know, okay, some experts may know already. They have a panel. You know, we had that with Laura. There were 2 panels, and they try to combine. Obviously, there it makes completely sense to cross-check with the others what panels you have and how we can optimize the 2 panels when you combine them.
I'm a massive fan of co-design. So particularly when you're working with threatened species, there's going to be a whole lot of experts in that area. I'm a geneticist, I'm not an ecologist. So partnering with the ecological experts and the on-the-ground experts is always going to be important. And they can tell you things like what are their management questions, what are they hoping to know, what's going on with the species on the ground, because that can help you look at the longevity of your SNP panels as well. And can help you come up with some simulations around, you know, are they doing translocations? How many individuals can they move between areas? What's their kind of population sizes they're trying to address? And can help you when you're designing your panel as well, you know, how many genetically distinct populations might there be and can you get samples from across their range? So all of that kind of co-design will come into it in your SNP panel design. Then, yeah, as Bernd said, in terms of the cross-checking, I mean, obviously taking them through the data that you've generated and showing how your SNP panel is likely to compare with that GL check panel function, showing how it compares for some of the metrics that they might be interested in would be important. Yeah, but the extent to which they can cross-check it, I'm not entirely sure.
Okay, Elise, you want to unshare your screen? Maybe close your mic. I can use mine. Okay, give me a sec to share my screen. Took me a week to work out how that works, and now it's almost over. Okay. No, I don't need that. So let's go through the tutorial, it's called, and we go there step by step. Hopefully you can see my screen now, and we run the first bit by running and loading the packages, yeah, and the data. If you do it like me, you know, I open the RMD file here, the path data is fine. If you run it by the tutorial session, you have to adjust that. And so maybe you're not sure, do I adjust it and to confuse people or don't I? I don't know. I leave it like that. And if it doesn't work, you put an inst tutorial W15 in front of it, and then it should work. Took me a long time to understand, but if I run that here now, this works, even if everything seems to be wrong. Okay, so we load our data, and I will tell you later what it is. Basically, our first data is our red fin blue eye data. And the first thing what we want to do is, yeah, what kind of panel choice do we have? What do we want to monitor? Population structure, relatedness, sex-linked panel. We haven't done that, but you could do that, obviously. How would you do that? You answer that by yourself, how you would ask Niana, how would you design a sex-linked panel? Yeah, exactly. So that's actually an easy one because you can identify the loci which are sex-linked. Beautiful. And then that's basically your panel. You can try to optimize that a little bit and basically use the sex-linked function and see how good it is to predict the sexes, and then subsample your panel and make it more difficult, less difficult in terms of numbers of sex-linked markers you use. But there should be, you don't need many for sex-linked markers, because the moment you have 5 that are linked to a female and you have them and they are reliable, that's more important, you basically can identify the sex. Correct? Pretend she's nodding. Okay. So make sure the first exercise, do we do any breakout groups or anything? We thought about it, or we just go through them?
I'll see how we go. Next week.
Yeah. What's the time? Yeah. Maybe we just say, so again, think about what is the panel for. Yeah. That's very important. That's key. And then we can start to do filtering and going into the workflow. So we assume maybe first one, we look for a population structure. So we want to think about FST values of panel. We want to design one that is meant to be good for recreating FST values as closely as possible to the actual data set. Okay. First of all, we do some filtering in terms of the individuals, because when you want to look for a population structure, it may make sense to not include all the populations, because some of them you only have 1 or 2 individuals. And even if the panel would recreate this population nicely, you're basically pretty unsure if that's representative of the rest of the population. So first things first, if you do something like FST reconstruction with your panel, it makes sense to look for samples which you have a minimum a number, and we decided here 10 is a good one. But feel free to use a different number for that. Yeah. And so we basically keep the populations that have at least, maybe, I'm not sure I want to explain the code. These are the POP20. You name the names of our, maybe go step by step. We do a table on the populations. So we have here our TT. So we know these are all the population, and number below is how many individuals we have in each of the samples. And you can see some of them we have only 10, and we want to have more than 10 for our sample. That's this one. So this little piece of code says, give me all the populations which have more than 10 individuals. And check that. Yep, these are the ones, and you could compare, for NW72 is no longer in there. Okay, and then we use that with a GL keep pop. In the pop list, we put all the populations that have this condition satisfied. And then we check that, because we never trust us. And you can find out, yeah, all of the populations which we have in the dataset at the moment are above 10 individuals. Good. Please kick in if you want to. If I have missed something, just to let you know. Okay. Next thing, obviously, missing data. You want to remove loci which are not informative. If you have lots of missing data in one of them, and one is here, an obvious one. If you have a population that has have a loci that has only missing data for one of the populations you want to identify, that's not a good loci. Yeah, because actually could be actually good. Doesn't matter. Okay, you don't want to have that, because you kind of could be obviously if you wanted a mutation in the restriction enzyme could be really informative for this population, but quite often you would think it's missing data, and you want to make sure you don't have missing data in that population. So we filter for all missing data by population. So we get rid of loci which are missing in one population completely, or SNPs are missing, or genotypes are missing. That's the right way to say it. Okay. Next thing, we want to have high quality SNPs. So missing data overall is now also a key. You want to have something, and think about it, you only want to have maybe 100 SNPs, so you can filter hard. Yeah, you want to have high quality SNPs, so nothing stops you to do a call rate threshold of 0.99. You could even do 1, and you would still probably have hundreds of SNPs you can choose from. Yeah, and nothing stops you to check that. How many do we have at the moment? Are we at 2? At the moment we are 6,000, and before we had 9,000. So we lost some, but who cares? Yeah. So the call rate filtering. What is the next important thing to filter? Minor allele count. Why is that? Elise, you want to explain that? No, just I'm happy, but okay. Minor allele. Yeah, obviously, if you have a SNP which is only occurring an allele which occurring once or twice, could be super informative, but on the other side, it's probably not good to differentiate populations, because maybe only one individual in a whole data set will have that, but not many. So it makes sense to also look for things which at least are occurring a little bit more often than once, and we decide here on 5. Yeah? And again, there is no hard number, but these are most likely SNPs. We are still informative, and now we are down to 6,800. Yeah, for those. And then that's an interesting one, because we basically, when you get Dart data, and it's probably true for most of the other ones, the length of the sequence you get back from your SNP. So every SNP is basically a longer sequence, and normally you get 69 from Dart, or even longer, 128 sequence information for each of the SNPs. If your length of this is less than a certain threshold, it's most likely that something happened with that SNPs. It's not of high quality, or so you don't want to have that, and therefore you filter also out for the trimmed sequence lengths. Why 29 and 30 is just,
Can people hear me?
Yeah.
This was more, it's not necessarily low quality data. You can still get really good quality SNPs. It's just where the restriction enzymes cut.
Yeah, my mistake.
But the reason we're filtering here is because we need to be putting primers in either side of our SNP. And so very short fragments aren't going to allow you to do that. Alternatively, you might be trying to blast this back to a reference genome. And so if you've got very short stretches, it might hit in multiple places, or you won't get very high confidence of it being a good match to that location. Yeah. So we just want to filter out the really short things.
Okay. That's much better explanation. It's actually correct. Mine was wrong, but okay. So again, this is a bit a different way to filter. Basically, this gives me an index. And if you look at index, this is a true-false vector. So for every loci that's larger than 29, you get a true, and for every that's smaller, it's false. And we can use that in the GenLite object quite conveniently. This is the individuals, and these are the loci are behind the comma. So we can say, okay, everything that's false is kicked out. So that's why this is a nice way. It's basically a very short version of a filter. Fun? Are we going? Okay, good. Yeah, and then we obviously we didn't lose many loci, but now we're at 6,600. And another one is alignment quality to a reference genome. So if we have a reference genome, okay, this is flickering here. So maybe if we're out of reach in a sec, then that means because we have a power outage. What you can do is if you have a reference genome, you can blast your SNPs against the reference genome, and you want to have one that has a high bit score. Means they match really well to the reference genome. You know where they are, and the sequence is actually really, you can trust it. Very important for your SNP panel, and therefore you want to have that. We're not running here a blast because it takes a while. It's the GL blast function, if you want to know. And you can see it you could see for a second, it has the arguments reference genome and your GenLite object, and you can put in some other values to look for quality. Basically, we do that later, so you don't need that. But now, how well it hits to the reference genome using, and here important again, because we filtered for the length of the sequence, at least 30, a sequence of 30, because we filtered for that before. So we get hopefully only SNPs which have a really, really nice hit. And we use the bit score for higher from 100. So that's, yeah, it's pretty. It's basically, there's no question that is the right hit, is it? And so once you have that, and we did that in the background, so you load this data set. I did before, but I can do it again. So we are at step number 5, and we can basically, again, create this index, use everything that has a bit score of higher than 100. And then you can filter again for that. And now we are down to 5,900 loci. Okay. And for some reason, I have to explain, no, I don't have to explain, you want to order your individuals and have to make sure by population, because when you do your SNP panel selections, this makes it much more efficient to run. So you want to have that, and in the future, this will be implemented in the panel select function, but it's not there yet. Okay. You want to do that?
No, I can't.
Yeah. So basically, the idea of this exercise was the following. You find it as in the tutorial. You can run obviously different filter settings. You know, you want to run for higher minor allele frequency, see how many SNPs you get, and that's the idea of this exercise. Because of time and so on, I think we skipped that, but it's important if you want to know. You play around and see which one of the settings of your filters are most important in terms of your quality for your SNPs. So you would try to play around a little bit with these values and see if this makes any difference. For example, obviously, if you can go to core rate higher than 0.99 or 1 and you still have 10,000 SNPs, why wouldn't you? Yeah. And the same for the other filters. Okay. Now we come to the fun part of it. Elise said there are 2 kind of functions we talk about, and they have some settings. For example, the GL select panel function is the one where you create a subset of SNPs, and we have different methods implemented. And we basically made them up over the time. We thought, oh, what is important? And I'm happy to try to explain some of them, but the other ones, you have to read the documentation. I'm a fan of this one, that's why I have to explain it. What is the idea? If you look, we thought about, okay, what would be a good idea to look for SNP that identifies structure? And the idea was, DAPC is a nice method, normally, because what it does, it finds SNPs and it orders them in terms of their ability to discriminate between populations. And I said, oh, that's cool, because obviously that's what you want. You know, you want those ones which are the ones which are very informative to separate these. Basically, in some sense, to make a PCA where all the dots are as separated as possible. Yeah. And here comes my brilliant idea. Let's do it in an even more sophisticated way, because we have, say, 10 populations in here. Why not using a DAPC between all the pairs you have in the population? So 10 times 9 divided by 2, 45 pairs. You do a DAPC for every of the 2 pairs, find out which SNP is best to differentiate between these 2 ones, and do that for all of them, and then combine take maybe 5 of them from this pair and 5 from the other pair and 5 from the next one. And in the end, you get a panel that should be really good in differentiating between populations, because you pick every time the best ones. And actually, in some simulations, it performed quite well. Elise has still the competition. Which one was your favorite?
PA high.
PA high was actually also pretty good, which is private allele frequency, private allele, but also the frequency. So you have alleles which are private for some population, again in pairs, and you take the one which are highest, and that's also pretty good. And it's kind of a very close race. Sometimes this is better, sometimes better. So the different ways to select panels, and for some of the things you're interested, they make sense from theory, deduction, armchair argument, why they should be good. What is another one? Monopop is basically things which are only occurring in a population, or kind of they're fixed alleles, which are only in one population. Obviously, they're good for structure, but you won't find, probably it would be great if you would find for every population which is it's only occurring in that population, then you are solved, basically, if you're interested in population structure, because you know exactly where they're from. Stratified is simply you sort your allele frequencies of the loci and pick one from every kind of basket. So from 0 to 10% in allele frequency, 10 to 20, 30 to 40. This was meant to be a method which just kind of gives you a good reflection of the diversity. And that could be a nice way if you're interested in diversity measures. PIC, we talked about that, is information content. Is basically the product of high in allele frequency, but also very evenly distributed across the population and individuals. So the different versions, in short, how you can select a panel, and I'll show you now how that works. You say a panel for DAPC, and it's maybe not a good idea to run that, but because it takes a bit, but let's do that. Basically, this says select 50 loci using this DAPC method on our complete data set. And now, after some time, you should get a GenLight object that has 50 loci, and they're meant to be good. And what do I mean when I say good? We can check the panel, and by saying, okay, please use the panel and the original kind of populations you had, and see how good is our FST value recreated. Should I wait, or should I show the answer? Give me another second. Are there any questions so far? Yeah, use sex-linked loci. Yeah, Jacob, you got it. Probably earlier than Diana.
There's one more question.
There's another question.
I'll just answer this question while the that's finished running, but that's all right. I'll answer this question. So for
done.
For the eDNA samples, how do you deal with the frequency calculations with multiple sources of DNA? So that's a good question. Obviously, when we're taking a water sample in a pond containing multiple fish, we're not going to get genotypes from individuals. So it's a different way of looking at it. So I'm actually going to be looking at population level genetic metrics. The other way to do it, though, is if you take individual fish and put them in a cup of water, or just have one fish in a tank and let it swim around for a little while, you can then take water samples from that, and then you can hopefully get at your individual genotypes. The genetic information that you get from your water samples, though, is eDNA is going to operate in very different ways to tissue samples, though. So I don't know if you work in the eDNA space, but the way that eDNA is sheds from individuals, the way that it accumulates and the way that it degrades can really impact those kind of frequency estimates that you get. So there's a lot of other layers that go on top of it for eDNA work. But yeah, I guess this is looking at how close can we get at those frequency estimates and how well do they match the reality when we're getting tissue samples from those individuals.
Okay, cool. So we got from our GL select panel function, we got what we wanted. We got 50 SNPs. Sorry. Okay, so we got back from our GL select panel function, and you can find we found it's in the object, which is now still a genlat object panel DAPC, I called it. It has 50 SNPs, and obviously we got it from all of the individuals we had in ours. Where we selected them from. And now the question is, how good is that? And we came up with this way. There might be better ways, so please be patient enough. We used R square values. It's probably not the one because you want to have also a measure of the variation around it. So maybe there's some other metric, but at the moment we have that. And it basically is very simple. It uses the panel and the original data set and makes a nice plot, hopefully. And if this should have run it, and then we can hopefully answer one of the questions with what do you mean with it doesn't reconstitute the original values. It's a new ruler, I think that's what Elise called it, and I show you in a sec. Is that slow today or is Emma? Okay, that's good. So that's what you get back. You actually get also a data frame, but that's the more important bit. Here is the original FST, and here are the FST from the panel, and these are all the different pairs of my 10 populations of 45 dots, I think, or was it more? Oh, I forgot that. How many do we have in hand? I call it panel A. 14, okay. So it's more dots. It's 14 times 13 divided by 2, whatever, 100. Yeah, potential pair. And you can see, that's important. The ranking is pretty nice. So the original FST were 0.1. If I used my panel, I get a higher value, 0.2. Yeah, but a population that has been kind of low in combination with a pairwise FST between the two populations has been low. Is also low in the other one. So we have a nice correlation of 0.93. So the panel is actually quite good to recreate the population structure in that sense. Yeah. And there are other ways to test it. You could also do a PCA, obviously, you know, for the panel and for the whole data set and check if they kind of are similar. It's just one way to compare the quality of the panel. What it doesn't do is that what Elise wanted to say, you had an original 0.1 in your population. You don't get a 0.1 back. You know, you get 0.2 as an absolute value in the panel FST. So be aware of that. But obviously, if you have this equation and you believe in that, you could try to reconstitute the original FST value back also in the future, because you have this relationship. Hope that makes sense.
And this one kind of addresses one of those questions that came up about the difference between populations. So you can see at the high end, the two populations that were most differentiated are still most differentiated with the new panel, and similar as you go down the line as well.
Yeah.
So that variation's maintained.
Yeah, but obviously some pairs won't be maintained. If the variation is, it's not perfect, obviously, it's a subset. Yeah, if I had used all 6,000 SNPs, that would be a one-to-one relationship. Okay, now let's do it. Okay, this was the DAPC, my favorite method. What would a random panel do? Yeah. So we select, the good news about random, it's fast. So we select 50 SNPs randomly, and then we check that panel. And you still has to calculate some FSTs, you know, it's not. And actually we have a faster function now with a new data model. It should be faster soon. And we can check obviously random versus DAPC. and see which method is better. And you can see it's not too bad, but random obviously is not as good as before. So maybe you could start playing a little bit. Okay, maybe I get away with 40 SNPs using the DAPC method. So I saved a little bit. And then you could do, okay, that's FST settled. The next one I want to know, okay, maybe I want to measure also heterozygosity. So you try to design a panel of heterozygosity, you get a gendered object, hopefully you have another 20 SNPs left, and then you try to combine the panels and see how good they are, because why wouldn't you use FST also if it was optimized for another thing? And you can combine it one and run it again with a check panel and see how good that is.
And I might just jump in for a second there and say that that random panel that we selected, it did just randomly pick 50 loci. So if you run that again, or any of you guys that have run it from on your computers, you might have gotten a different data set. Sometimes, just by chance, the random panel that you select may actually be a really, really, really great fit for FST, but sometimes it might be poor. We actually have a poll to put up as well. You can just kind of get a feel for across the 50 or so panels that might have been run.
Sorry, that was the wrong one.
Just so we can get a feel whether random ever performs better or how much better your FST panel might have done. So if you have a look at the R-squared values that you got for your panel.
This is playing tricks on me. What's this one? No.
This one.
Yeah.
So have a look at the R-squared values that you get for your panel when you ran the DAPC method, and then have a look at the R-squared values that you got when you ran your random method, and let us know if your DAPC was better, and if so, how much better were those R-squared values, or was your random one better? Ooh, someone got a random one better.
Shouldn't have. Can't be. Obviously, the random one could be exactly the same like the DAPC just by chance. And here comes the thing that I think Florian was asking that. In the best of all world, it would be quite easy. We use all combinations of SNPs, 50, and see which one is best. Unfortunately, this is, if I'm not mistaken, more combinations than the numbers of atoms in the universe, as we always know. So you can't physically do that. So you have to find a strategy to find good samples, good SNP panels. And obviously, in some sense, it's most likely not the optimal, but it's a pretty good one, you know. And you can see, at least I had a random now one that's 0.81. And here comes also, nothing stops you to run a loop and play with that and use different combinations. You know, here it comes nicely if you use a little bit of R. Okay, let's try 40, 50, 60, 70 SNPs, use this method and this method. And we actually did that in one hell of a simulation. Do you have the slide? Because I couldn't find it. But yeah, maybe we show it here and then we go through the next part of it. So we did that, try to... Yep. In a simulation where you try to identify what is a good panel for this data set and run a simulation and explore the parameter space, and came up actually quite a nice one, which fits 4 of the 5 kind of things. We could never get NE estimates, which are good, but we could get other estimates quite nicely.
Frozen on me, sorry.
Okay. Maybe it wasn't a good idea. Random number generator, yeah. You could set the seed, yeah. But you get a panel back. Doesn't matter what your set seed was. Sorry, maybe guys don't hear what we talk. Do that later, okay? Yeah. So here, for example, we run a pretty comprehensive simulation. Doesn't look like, but we used all the different methods, as you can see. In the legend, we used FIS, FST, heterozygosity, and numbers of alleles as a output. And we compared, for example, the first row is a DAPC method. And you can see it performs beautifully for FST as predicted. But and it doesn't matter how many loci you're using. Obviously, 300, you almost get everything back except for inbreeding coefficients. They're really hard to get back. So even 300 are not bringing back the original inbreeding values. But you can see here, if you do the simulation, yeah, I get away with easily 50 SNPs for my FST panel. So maybe I have a place for 200, and I use the panels I got here for numbers of alleles. You can see the PA high method is pretty good. So maybe I use that panel for estimate the numbers of alleles, another 50. So you can combine them. In the best of world, you have a nice overlap. You even save some of them because they're the same. Yeah, so that's the idea. If you want to make it work, we try to really develop a pipeline with that, but we're not there yet. But I think the first step is there to try to identify a panel. And for some of the tasks, it's quite easy. That's the good news. What's the next one? Oh, yeah, that's the exercise.
Share that.
Oh, yeah, sure. You're getting towards the end anyway. You should check if there are more questions coming up. But I run through the final challenge. We had planned, so we give you an idea. You basically choose your own method. The numbers of SNPs you have, maybe you can afford only 20. Use it, select and check panel and find out, get an idea how many you want. And obviously use your own data set, not necessarily the one we gave you for the red fin blue eye. And this was, I'm not sure on this, unfortunate. It doesn't run. Does it show in the... I thought... Oh, okay, there should have been an output here. And it will take a while. Basically what this does, it runs for, yeah, we replicate for each of the panels. We have 20 replicates. We select 50 individuals using the method DAPC and run it in a loop as often as we want, 20 times, and see how good it is every time when you select using the DAPC method for a different subset of the data. Yeah, so we can, and you can do that as well. Basically, make sure this method is, you're not lucky, and you can then pick the best one of all the outcomes, and that's my panel. Okay, so you wanna wrap up? Happy to do so. Okay, first of all, when you select a panel, define what it is for. Is it for FST? If it's for heterozygosity? Is it for ID of individuals? Is it for sex ID? Or is it for sex link? So this is really important, or a combination of them. Then make sure the SNPs you have where you want to select from are really of high quality, not many missing data, high call rate, occur at least a certain amount of time, and if you can, use also the filter which are linked to a reference genome. So the quality you hit to the reference genomes, and make sure you have the information so you can put in a primer left and right from where your SNP is. And make sure you have actually a good even sample size from the populations you want to create the panel from, because it doesn't make sense if you have only 3 individuals in one population, 50 in the other one. You develop a panel which is basically only looking at the one where you have the higher sample size. Then once you have a panel, obviously go to your provider and make sure they can actually work with the amount of SNPs you have decided, and they have uniqueness in the alignment, and they're not interacting, which is also a problem in lots of SNP panels. And then you find them, and then you can test them. There's one more thing which I'm not able to show you, but in my previous session, the simulation sessions, we did actually have a simulation of SNP panels. So we selected a panel, and then we ran the population 20 generations, and then we applied the panel again, and we compared, is it still good in 20 generations to show me the ranking in FST values. And FST was easy. Some other things obviously are harder. And if you have something like inbreeding and you want to monitor inbreeding, but inbreeding is really strong, maybe your panel is no longer as good as it was. I'm not saying this is a definite answer because you simulate what's happening, but it at least gives you an idea what can I expect in terms of variation and how well will be my panel in whatever 20 generations. I would stop here. Questions? Yeah, there's a red dot. Unanswered. There's one. When selecting candidate SNPs for ESU identification, would you still verify the subset is similar to full dataset by comparing FST or PCA? Is a better way to verify in that case. ESU. I'll try to get that. What is a good way to comparing? Yeah, I don't have a good answer yet. You know, the R-square value is nice to give you how good the relationship is and how tight it is, but it doesn't give you a good measurement how effective the absolute difference is between your panel and the actual one. So there are better statistics doing that, but I haven't looked into that. But I would think R-square value and a PCA is a nice start. You know, if you can recreate the structure pretty nicely, and you could, there's something like Procrustes tests, but which compares basically PCAs to each other. I'm not a real fan of them because yeah, I don't know. You get a number out of it, but as always, you do a test and it's significant and it's not really, you know, it's more like in the management, you have quite often, okay, I want to know at least these guys are different to those guys and want to monitor that. And you look at that and you don't need a Procrustes test to tell you that if the panel is able to differentiate those two populations or not. And I knew Bill would say something.
I think maybe this person's asking if I'm wanting to look at gene diversity, would I use the FST comparison you've shown and the answer is no.
Okay, no. If that's the question, that's true. If we look, we haven't shown that, but you obviously can use also select a panel for heterozygosity. Yeah, so you can, maybe I should have, I think I skipped over that. When you say in the GL select panel function, yeah.
Do you want to share that?
Oh, yeah. You can, hang on, I share that. This is a convenient function. Obviously, you can use any measurement you like, but it's coming up, hopefully, no? Yeah. So the GL check panel function has not only FST, it has also other settings. If you want to compare, how good is a panel to compare observed heterozygosity, you can put in HO and then it calculates observed heterozygosity and see how good is a panel for this outcome. If it ever comes to some answer. Yeah, so you can use different parameters in the check panel function. So it's observed heterozygosity, expected heterozygosity, numbers of alleles, Did I miss something? I think that's it. Effective population size is in there as well, but yeah. Does it make sense? I'll stop sharing. Answered. There's one more, is there?
I was just answering this one in text, but just for other people's benefit. So how do you ensure that the water, I was talking about getting one individual fish and putting it in a cup of water or something, and then sampling the water from that to get individual genotypes from eDNA samples. And so there's questions following on from that saying, how do you ensure that the water that you're using, that you don't have DNA of the fish that you put into the water that can then contaminate your sample? So my answer to this was just saying that you don't necessarily want to go and scoop the water from the pond that has hundreds of individuals, because you're going to be getting DNA from hundreds of individuals in that water source. So you want to use some other form of clean water. And when I say clean water, I mean water that's free of DNA of your target species. Ideally, we'd use sterile lab water or something, but fish aren't going to be able to cope in those conditions because they're used to the particular water quality that they've been living in. So it's a bit of trial and error, well, not trial and error, it's a bit of thinking about your study species and what they're able to handle and what kind of water sources you might have nearby that would be similar to the water source that they exist in, but it's going to be free of DNA from your target species. So for the redfin blue eye, for instance, there are a bunch of nearby springs that didn't contain the species. So I was able to get water from those nearby springs that were free of redfin DNA, and then put the individual redfin blue eyes into that water.
That's definitely answered. Thanks a lot.